Welcome to today's edition of Conversations on Ocean Carbon, a U.S. West Coast and Alaska perspective. This webinar series aims to introduce concepts related to ocean acidification, hypoxia, and other marine carbon issues. My name is Alex Harper. I'm with the Central and Northern California Ocean Observing System and coordinator for the California Current Acidification Network. I'll be today's moderator. The title of today's webinar is Introducing the California Ocean Acidification and Hypoxia Portal, a centralized information hub to enable synthesis and understanding of statewide status and trends. I'll be joined today by Lauren Linsmeyer with the California Ocean Science Trust. Today we'll be conducting a few poll questions to learn more about the audience and interest in the California OAH portal. So Emma, who's working behind the scenes with Ali Boutros from Sencus, can we go ahead and launch this first poll question? Thanks. And so we're just asking for your primary affiliation, again, to get a sense of who's interested in this work, who's participating on these webinars, and how we might look to develop webinars in the future. Thank you. So a good spread of participants. Today's webinar will run approximately 60 minutes, and we're recording and we'll share the recording after the webinar. We will start the webinar with the California OAH portal demo led by Marine Lebrec, technical manager with the Central and Northern California Ocean Observing System, and Iwen, technical product manager with Axiom Data Science. Following the demo, we will have a short Q&A. Then we will hear from some of our project collaborators, including Christina Frieder with the Southern California Coastal Water Resource Project, Jan Newton with NANUS and the Washington OAH Center, and Erin Satterthwaite with California Sea Grant and Cal Coffee. Attendees will be in listen-only mode, and you can type your questions into the Q&A function at the bottom of your screen. We will post them to the speakers during the Q&A session. Before we begin the demo, I'd like to give a quick background of the California OAH portal project, its inception and development. The California OAH portal was supported by the California Ocean Protection Council and California Sea Grant, with the goal to build a centralized information access hub to serve automated, high quality, and interoperable data and synthesis products. The primary audience initially for this work was state managers, but project leads quickly recognized the value and importance of this effort for many other groups, including shellfish growers, water quality managers, data providers, researchers, and other community members. This work builds on many decades of efforts by the ocean observing systems to deliver high quality oceanographic information on trends and changes along our coastline. A lot of this work was done by folks like Marine and Iwen behind the scenes to standardize and build automated pipelines to deliver existing ocean acidification and hypoxia information. This work also builds upon the California Ocean Observing Systems data portal, calus.org, which I encourage you to visit. This effort builds streamlined access to existing high priority ocean acidification and hypoxia data, including from PMEL's West Coast OA cruises, and built novel approaches to exploring and visualizing that data. We worked with collaborators like Dr. Tessa Hill from UC Davis to streamline access to synthesis data sets, such as this multi-stressor observations of coastal and hypoxia acidification, representing over 13 million observations, which are now available in the portal. We worked with novel observing systems such as the Trinidad Head Ocean Observing Node, or THUN, which was established through the same project funding line, Prop 68 in California, and worked with stakeholder groups such as water quality managers and oyster growers to build dynamic dashboards and other information products. In the portal demo, which will commence just after this talk, you'll see how data sets can be explored and visualized. And our talks at the end of the webinar will explore synthesis products to understand trends and changes over time. And without further ado, I'd like to switch to the portal demo and perhaps launch another poll question while we transition to Iwen's presentation. So this next question is about how you regularly access information about ocean acidification and hypoxia. Mainly publications, web platforms, and webinars. Great. Thank you for that feedback. All right, Iwen, over to you.
All right. Thanks, Alex, for the introduction. And hi, everyone. I'm Iwen, as Alex mentioned, a technical product manager at Axiom. We are part of the Tetra Tech Ocean Science team, and we work closely with several IUS regional associations, including SENCUS and SCUS, to support their data and information management needs. So I'll be kicking off the portal demo today, starting with a general overview of how to navigate the portal and find information of interest. Then I'll pass it over to Maureen to showcase some of the key data assets we've integrated as part of this project and how you can explore them on your own in more detail. Let's start from the portal homepage. So scrolling down, you can read about the portal, get immediate access to real-time water quality conditions in California, check out some of our featured products that highlight ongoing OA monitoring programs, such as the West Coast OA cruises and the California Current Ecosystem Project. And you can access additional resources that are linked down below. So there are four main ways that you can search and explore data that are in the portal, and you will always be able to access them using the navigation bar at the very top. The catalog, the map view, the real-time dashboards, and the data views. Let's say you're most interested in current water quality conditions. The quickest way to find that is through the dashboard. So let's go look at the real-time pH conditions using the quick link on the home page. Here you've got current pH conditions over the past 2 weeks at coastal monitoring sites from San Diego at the southern border all the way up to Humboldt at the northern border. Hovering over the circles on the map will allow you to see what additional data is being collected at each location. You can also switch the map view to look at salinity up and down the coast or oxygen instead. But perhaps the statewide view is beyond the scope of your interests. Maybe you only need to see conditions at your shellfish site in Central California. If you click on the Station tab over here, you can select a specific site of interest. Let's look at Morro Bay. You can now quickly compare conditions at just this site over the past 2 weeks. Alternatively, you can also go to the regional dashboards from the drop-down in the navigation menu. This will show you a more comprehensive list of measurements, including weather data such as air temperature and wind. It's a little bit laggy there. So for access to data that's further back in time, you can always click on any of the site names in the dashboards and throughout the portal, which will take you to dedicated station pages where you can explore all the historical data at that site in more detail. But now I'd like to know what additional ocean acidification and hypoxia data is available beyond the coastal monitoring sites that we just looked at. So let's go to the main catalog page to find more ocean acidification data. The data included in the catalog has both recently integrated ocean acidification data assets, as well as data we've already pulled into our centralized system over the years, such as the MER satellite sea surface temperature dataset. Having these public data in the same place enables users to look at and compare assets in a more accessible way, which you'll see an example of how to do this a little bit later. It looks like there's almost 1,000 different data assets in this catalog in 3 different categories. So some data assets are called sensor stations. These are your fixed buoys or coastal pier monitoring sites collecting a wide variety of ocean and atmospheric data. On the right-hand side, you can scroll through the variables list to get a quick glance of what is being measured. Moving platforms, on the other hand, are typically data collected on a trajectory, such as gliders and cruises. Then you've got the data layers, which are everything else, including models, satellite, and compiled data from long-term ecological or cruise-based monitoring programs. If you are looking for data from a specific data provider, you can scroll through the list on the right over here to see what is available. So clicking on one of these applies that filter to the search. But you can always reset your search by clearing the filters at the top. If you're not completely sure where to start, you can always look at the list of tags on the right to identify a theme of interest, which can be helpful. I'm going to click on the ocean acidification and hypoxia tag. This will pull up key ocean acidification assets that we recently integrated as part of this project. Alex showed you the multi-stressor observation synthesis dataset earlier. We've got the West Coast ocean acidification dataset and ROM-spec. I'm going to clear this search bar again, because you can, of course, do a text search directly if you know exactly what you're looking for. So searching ROM-spec pulls up these search results. You can click on the result, which will pull up the catalog page for this asset. All the data layers in the catalog typically have a brief description of the data set, some information about attribution, source, and usage, as well as a list of the data layers that are available. From this page, you can find data access options, or you can add the data layers to the map directly for further exploration. Let's add the nitrate layer to the map and navigate to the map view from the navigation bar up top. So for most new portal users, there will be a couple default data layers that are queued up, the CalHab map and the ocean acidification and hypoxia sensors. For now, I'm going to remove these and show you the general, how to generally navigate the map view. So while this one is loading, it looks, I've got my nitrate parameter selected over here. If you click on the drop down next to the nitrate, you can easily toggle through and look at the different parameters that are available for this model. right, so that's lagging a little bit. Let me see if I can refresh and have that show up. All right. Well, that's unfortunate. Well, what I wanted to show you was that mostly go over the legend where you can adjust the color ramp, some filters where you can see model data down to 1,000 meters, and you can adjust the time filter down below. But Maureen will show you some other data layers that can demonstrate that example instead. So maybe let's hop over.
That sounds good, Ewan. Yeah, I'm guessing it has to do with having the Zoom webinar running while you're trying to navigate the portal. And I know you have to have your video on.
Yeah, I think let's
This
switch over to
software.
Maureen. This is a good spot.
Okay, Maureen, do you like to join us and share your screen?
Yes. Hi, everyone. Okay. Can you see that okay?
That's good.
All right. Thanks, everyone, so much for being here. My name is Maureen Lebrec. I'm a technical manager with the Central and Northern California Ocean Observing System. And I've been working on this OAH portal project for the last several years, working with Axiom Data Science as well as data providers across California and across the broader region to get their data into this data portal. So what I wanted to do is highlight a few more data sets and data layers that can be visualized through the data portal. And then I'll walk you through ways to compare data sets to each other in a tailored data view. And so I'll walk you through that. And so to start off, I wanted to highlight this MoCA data set that Alex mentioned. So this is the multi-stressor observations of coastal hypoxia and acidification synthesis product. This is a data product developed and published from folks at UC Davis and is a compilation of over 13 million observations from a number of different monitoring programs across the West Coast. And so the way that I can access this data set is by, within the map view, clicking on this Find Data tab, and I've just searched for MoCA. From there, I can add it to the map by just clicking that plus icon. If I then go and hit the legend there, we can play around with the map view a bit more. So I can change the parameter that we want to look at. Say we want to look at water temperature. I'm going to zoom out a little bit and you can really see the spatial extent of this really massive data set. Another really useful tool is being able to filter by depth. So this is right now looking at data from the surface all the way to 5,000 meters. But let's say we were interested in, say, the top 150 meters or so. I can use this slider to filter that depth variable. From there, we can also look at other variables. Let's say we wanted to look at pH measurements. We can access pH that way. Something that you can do as well is change the color palette. So let's change it to something a bit more useful to look at. And then what I'm going to do is zoom into a region of interest. So let's say we were interested in looking at pH from the San Francisco Bay or just offshore of San Francisco Bay. One really useful tool within the data portal is being able to aggregate data from a number of different points into a single time series. And so the way that I'm going to do that is by clicking on this polygon tool here in the top left. And from there, I'm going to draw a polygon around my area of interest. Once I've done that, you can see that it's created this time series of pH from all of the points within the polygon. And I can change the time bin to something a bit higher resolution. So let's say we wanted to look at weekly pH from this entire time series. And now we can see that that pH time series at a much higher resolution from 2011 to 2020 from all of these points within that polygon. Another really useful aspect of bringing in this MoCA data set into the data portal is being able to filter by data set name. And so if I click on that data set name, you can see all of the different data sets that went into the synthesis product. So if I click on this first one here, this is just showing now pH from these access cruises up here off of San Francisco Bay. Okay, I'm going to switch gears real quick and show another data layer, which is the California OAH, California Ocean and Acidification and Hypoxia Sensors layer. So I'm just going to add that to the map. If I navigate back to the legend, we can filter by pH. And so this is showing observational data from fixed platforms that are measuring pH up and down the coast. If I hover my mouse over a particular point, I can see the most recent measurements. So for instance, this is the Monterey Bay Aquarium seawater intake showing pH, and other variables just within the last couple hours. If I was interested in looking at the full-time series from a particular site, I could click on that. So let's say we wanted to look at this California current ecosystem mooring. I've just clicked on that mooring, and here we're looking at pH from that region, from that mooring. If I click on the legend here and remove the seasonal statistics just to clean up the plot a little bit, and then I can change also the time bin to something a bit higher resolution. So within just a few clicks, I've been able to create this plot of pH from about 2013 to 2022 from the CCE mooring here off the coast of Southern California. Another data set that I wanted to quickly highlight is just showing ways that we can visualize biological data. And so I'm going to search for the term zooplankton here in the Find Data tab and add this CalCoffee zooplankton biovolumes data set. So this data set is a sort of a proxy for zooplankton biomass and includes data going back to the 1950s all the way to near present time from the CalCoffee cruises. And so getting this biological data into the OAH portal was really part of a broader effort to be able to visualize and compare data from co-located chemical and biological observations to better understand impacts of OAH on the biology that we observe. And so from the portal, we can change the variable. So there's two different variables that are part of this data set. We can also filter by the cruise line. So if we know that we're only interested in this northern line, for instance, we can filter that way. And then similarly, I can use this polygon tool to create a time series of the zooplankton biomass or biovolume from this entire period. Similarly, I can change the temporal resolution to something a bit higher. And again, within a few clicks, we've been able to access this wealth of information. Okay, so I'm going to switch gears real quick. And what I wanted to do now is compare data sets together in a tailored data view. And what I'm going to try first is seeing if that ROM-spec layer is working. And if it's not, then we can just use another data set. And while that's thinking, what I want to do is compare two different data sets together. So to do that, let's say we wanted to compare time series of water temperature from one mooring to another mooring or to a model. So to do that, what I'm going to do first is click on the CCE mooring, and I've pulled up the water temperature time series from that mooring. What I'm going to do next is click on this little star icon. What that does is it will add this time series and data set to a data view. So I'll show you what that means. So if I click on that, I'm then going to add a new data view, and I'm just going to call it CCE1 or CCE analysis. Okay, I've clicked on the plus icon, and then I'm just going to check Save to Data View. Okay. What I'm going to do next is compare that time series with the other CCE mooring, which is located farther offshore. So let's click on this farther CCE mooring. And I'm just going to get a time series of water temperature. And it looks like it doesn't have that much of a time series, so maybe that's not the best one to use. Let's pick maybe the Monterey Wharf or this M1 mooring, which has a longer time series. So I'm going to just click on time series, and Similarly, I'm going to add it to my data view. So I'm just going to check data view here. Okay, now I can navigate to up here where it says data views, and I'm going to click on CCE analysis. Okay, so now I've basically created this data view, which is sort of like a tailored dashboard where I can visualize multiple time series together. Okay, so up here we have the M1 time series, and then down here we have the CCE time series. What I want to do now is compare them together. So I'm going to click on this Add to Compare chart, and I'm going to do that for both of them. Okay, so I'm going to just use this slider here to be able to see the time series, more fully. And what we can see is now we've created this figure with both of these moorings overlaid with one another. So this is just a really useful way to be able to compare, multiple data sets, multiple time series together. And the last thing that I want to share is that anyone accessing the OAH portal has access to these default data views. And so if I click on this NOAA PMEL CCE moorings data view that you should all have access to, you can see what a kind of final data view looks like. You can add some custom text images. And in this case, we were comparing pH from the coastal mooring to the more offshore mooring and being able to tell a story with that data. I'm going to pause there. And the last thing that I'll just mention is that up here in the top right is this send us your feedback button. So if you click on that, you can send any comments or feedback that goes directly to myself, Iwen, the rest of the data team, and I encourage you to use it. I'm going to pause there. Thank you.
Thank you so much, Maureen. Great overview. You covered a lot of data and a lot of functionality in that short time. So thank you so much. I want to get over to our three speakers for today. But quickly, a question for you and Ewan from the group around data quality. And so can you talk a little bit about any assurances built into the portal around data quality control or flagging, maybe in one of the station pages if you want to show where that information is viewable.
Sure, let me share my screen again real quick. So Ewan showed a really nice way to access shore station data through the dashboards here. So I'm just going to use that to access some data from just a single station. Let's just look at this Humboldt Bay Pier site. And so one really useful way to access quality control information is through a method called QORTOD, the Quality Assurance for Real-Time Oceanographic Data, which is a protocol that is used across the entire Integrated Ocean Observing System network, and is incorporated into the data pipelines that we have for getting these data into the data portal. The way that you can access that information, or one way that you can access that, is through when you're on a sensor page, you can click on Sensors with QC. And from there, you can access quality control flags. And so if I'm just going to zoom into this area here, you can see that there's instances where the green is data that have passed the quality control flags, but then certain areas have some pass and fail instances as well. So that all of that information is accessible to users through the data portal.
Thanks, Maureen. And you'll see a quick question from Greg in the chat around whether the data is downloadable and in what format. So this might be a good place to sort of show where you can download and the formats.
Sure. Yeah, if I go back to the sensor page, there's several ways to download data. One is by directly clicking on this downloads button, and from there you can access data in a number of different formats, including CSV and NetCDF. We also serve our data through an ERDAP server, and you can access that either by clicking on this button here. There's also this ERDAP station page listed here on the left. And if I click on that, it will bring me to this ERDAP server where I can access all of the metadata information about that particular site. I can also download data and subset data through this ERDAP query page. in a number of different formats, and you can see all of the different formats listed here. So a lot of ways to customize how you're getting that information. Yeah, great question.
Yeah, thanks for running through that, and don't hesitate to reach out offline if you need any assistance on this. Okay, let's keep moving for the sake of time. We have one last poll question about having explored the portal a bit, whether you're likely to use it in your work. And as we get that poll question launched, we can move over to our first speaker, Christina Frieder from the Southern California Coastal Water Research Project, or SQUIRP. She's going to be providing a talk about utilizing the OAH portal that we've just seen as a visualization tool for numerical ocean model outputs.
All right, thank you, Alex. Yeah, so here I'm showing, this is what the portal would have looked like if it loaded, but this is an example of model outputs for aragonite saturation state from a single depth layer, and how much you can view in terms of trends in ocean acidification, both in space, and then also there are some views to look at patterns in time as well. The model that we're working with and sharing the outputs for is what we call ROM-SPEC, or the Regional Ocean Modeling System, coupled to biogeochemical elemental cycling, which means it includes an online biogeochemical model that has a lower ecosystem function. So things like phytoplankton groups and zooplankton are incorporated into the model. This model has been under development for over 10 years in partnership with University of California, Los Angeles, Princeton, as well as our modeling team at SQUIRP, largely through funding, again, from California Ocean Protection Council, as well as NOAA. And the reason, one of the impetus for developing a model for the US West Coast was to understand what is driving ocean acidification, oxygen loss, and harmful algal blooms in this system, in particularly, not only from natural climate variability, but that from climate change and that from eutrophication. So our land-based nutrient inputs that are going into the coast in the Southern California Bight, along the San Francisco Bay and Monterey regions, and what is the relative contribution of eutrophication versus climate change to changes in ocean acidification. There's multiple domains for which this model covers. And so the ones that we've uploaded to, the outputs that we've uploaded to the OAH portal are those within the blue bounding box, which covers almost the entire California coastline. The primary ways in which you can access interpretations and synthesis of these model outputs has been via publications. And there are a series of publications from ROM-SPEC on ocean acidification. And the model's been really well validated with observational data across all these domain scales, even for relevant ocean acidification variables. But if it's only available for publications, that makes a real challenge for other users or interested researchers, industry, or water quality managers to access this information as well. And so that's been a real challenge in terms of working with model outputs. This model itself, it's mechanistic, and it has dozens of variables that are predicted across millions of grid cells, both horizontally and vertically, on time steps of approximately 30 seconds. So you can imagine how this results in extremely massive output files that are in specialized data formats. And so it really requires technical expertise and able to visualize them, make an animation like this to look at patterns in space and time and ocean acidification variables. And so we wanted to work with the OAH data portal as an avenue for which to visualize the model outputs. So what's available is a 20-year hindcast at daily resolution, and really allows you to visualize dynamic circulation features. You can see upwelling dynamics and how that drives changes in carbonate chemistry variables. And the information, it's not just surface maps or single depth layers, but you can also click on any location within the model domain and see how the trends at that single location. So here's just a snapshot of what that might look like. If in the OAH data portal, you click on a single location, you can now see, this is an example, Hofmahler of aragonite saturation state with depth and time, and even a single profile from an individual week. And you can see the depth at approximately 50 meters, by which aragonite saturation state is already falling below that relevant numerical value of 1.0. So this is just kind of an example of giving access to people, easier access to play around with model outputs than has traditionally been available. Of course, this is just, this isn't really synthesizing the information. And so really, Faisal and myself and the rest of the modeling team at SWRP are really looking for feedback and information about what are curated views and synthesis products that would be useful from these model outputs. So here's just one example. Maureen walked through an example curated data view from the CCE moorings. We also have a curated data view of aragonite saturation trends at 3 locations along the California coastline. So there's southern in orange, central in green, and northern California in purple. And along the entire coast, there are these long-term trends of a decrease in aragonite saturation state on the timescale of decades. It's consistent across the entire coast, but you can really see that the underlying variability is much different among these 3 locations. So this has been our first pass at providing a synthesis curated product, but we're looking for feedback for what else would be useful and insightful to the water quality community. And also I know that the next 2 speakers are going to focus on how you even translate this information a step further of not just what are the trends in chemistry, but how can you make interpretations from a biological perspective.
Thank you, Christina, for that great overview. Perhaps Maureen or Ewan can drop a link to the ROMSBEC data view that's available in the portal for folks to explore and perhaps provide feedback on. We'll switch to our next presenter now, Jan Newton from NANUS and the Washington OA Center to give a further view of how this information and data can be supportive of a ocean acidification indicator for the US West Coast and California. Thanks, Jan. Take it away.
Thank you, Alex. Thanks for the invitation to be here. So I am representing a team that has been engaged by the West Coast Ocean Data Portal to serve the West Coast Ocean Alliance and funded through SWRP to develop indicators for ocean acidification. And what I'm going to talk about today is the application to California. I want to acknowledge the team members, Kate Hewitt of the University of Washington, Christina, who you just heard from, Adrian Sutton and Simone Allen from NOAA PMEL, the lead scientists on this project, and then also aided by Hannah Bussey at UW and formerly Roxanne Carini. All right, so let's tell this story. So indicators, you're hearing a lot about indicators, and they're so important because we have these data. What we need to be able to do is to communicate that well to people who want to understand this status of things. So we had this process for the West Coast OA Alliance, and this started in 2022. There were consultations with 29 state, tribal, federal scientists, including the USAs and managers in California, Oregon, and Washington. The working group that I showed you just previously. We're currently in phase 3, and this is for the whole West Coast, which is to produce the 2-pager and a website, and that's due the end of September. So when we heard of California Ocean Protection Council's interest in an OA indicator, I was personally thrilled that OCP will mirror the same approach and produce not a separate indicator, but one that is specific to California, but is benefiting from the several years of co-development. So these are internally consistent. All right, so I'm going to focus a little bit on the West Coast Ocean Data Portal OA pilot indicator development. So the objective for that, you can read, the intended audience is high level. The purpose is to provide that big picture context and education about OA status and trends to increase awareness in support of policy and decision making. All right, so that was our marching order. As we spoke with the 29 individuals, I should have found one other person so I could just say 30. 2 questions emerged. What do you really want to know? What do you need to know? And so one of the questions is, how different are conditions? How different are conditions now versus some reference level, which we chose to be pre-industrial conditions? So that was an important question. The second one was, why are these differences important? What is it going to do to biology that exists in the ocean? And so even though we were going to use chemical data, we were going to view the chemical data through a biological lens. And when we first started out, we were thinking of a species-specific threshold and talk about the duration of the record for which current conditions cross that threshold. But as this has evolved, we're more using the model data to evaluate habitat that's corrosive at aragonite saturation state less than 1. But it is flexible to, find different values if you do want to take the species-specific approach. All right, so let me explain that a little farther. All right, so here you see the 2 indicator questions, anomaly from a reference level, pre-industrial, and some biologically relevant chemical threshold. You can make an indicator based on observations or based on models. And we chose to primarily focus on the observation to answer the first one and the model to answer the second one. But as you'll see, both of these data sets and these indicators are useful for both of these questions. So why are we really focusing in on the climate, on the observations? Well, it's that word, climate quality buoy data. If we're stating something is different than pre-industrial, we really want to have our best information on what that value is. Why use a model for the biologically relevant chemical threshold? Unfortunately, our buoys are primarily surface values, and so a model gives us the full water column, and so was able to assess the percent of volume of the shelf waters that were corrosive. Okay, so that is our aim. All right, so let's talk about observing data. We, first of all, congratulations to the OAH data portal team. That's a great tool. Back 3 years ago, we accessed the West Coast OAH monitoring inventory and asked for data that had 2 carbon variables and were recorded with time. So not that many compared to all of the huge numbers of observations. And then when we started getting down into the details, getting at the climate quality, we decided to start with the gold standard, which is NOAA PMEL's ocean acidification program, NOAA North National OA program. Anyways, the national buoys that are funded through the NOAA OAP ocean acidification program. There are 5 of those along our West Coast, 2 in Washington, 1 in Oregon, and 2 in California. And Maureen mentioned one of these buoys. All right. Adrian Sutton is the lead for these buoys and had published how to use present-day observations to detect when we were outside of pre-industrial bounds based on the carbonate chemistry. This is a published paper in 2016, and they have these plots on the PMEL site where you can interact with that and see how far out of the pre-industrial bounds we are. And we can see great regional differences. We chose to follow this approach. As I said, when you're going to start something, start with the data that you're most certain about and that has been peer reviewed as terms of its method. So these are plots from Adrian, and you see 2 different patterns here. The first one is the percent observing days when conditions exceed pre-industrial variability. So that was that first question that people wanted to know. And it's color coded, the percent, the darker the color, that's a higher percentage of observing days exceeding pre-industrial variability. So you can think bad. And it's interesting because you see in the Southern California current, you see some really dark numbers, including meaning higher percentages. So that's interesting. And there's somewhat of a gradient north to south, but it's interesting to see those higher values in the Southern California current. Then when you change your question to, well, maybe my organism doesn't really care about pre-industrial versus not. Maybe they just really have this threshold. And we used, instead of 1, we used 1.7 since these are surface values only. And then you see that the percent of days that conditions were less than 1.7 omega aragonite is much less in Southern California. So with any indicator, it's really important that you think about what question you're asking. And what I'm so happy about is that while we're producing this 2-page indicator, document, there will be an accompanying website where people can go for more detail. But to indicate the trend, we chose this percent of observing days exceeding pre-industrial variability. And I'll show you what that looks like in just a moment. Okay, so then to assess what we can learn from the models, we use not only ROMSBEC that Christina introduced so well, but also for Washington and Oregon, the live ocean model that Kate Hewitt and Parker McCready have developed, and Kate has done the work for this project. So we use those 2 models to assess the cumulative corrosive volumes across the regions and across years. We did choose omega aragonite saturation state. We chose a value of 1 for this since we're looking at the full water column. And we're looking at then how they compared with the pre-industrial estimates. And that was a difficult thing to do, and we've done it slightly different for the 2 regions because of some of the idiosyncrasies. So this is how we are assessing status for the 2-page indicator. So this will make a little more sense as I continue onwards. All right, so here is all of our work boiled down to the status indicator. Now, ROMSBEC, as Christina introduced, has been run through 2017. So the status that we are able to present right now is for 2017. And so on the scale that we developed compared to pre-industrial, that receives a fair. Once the ROM-spec model is updated, we will update this status as well. But for now, that's where we are. Shifting to the trend, this is the percent observing days conditions exceed pre-industrial variability. This is the plot from Adrian showing the South California current offshore buoy in blue and the Point Conception buoy in black. And one thing I want to draw your attention to is that 2017 was a very different year than 2024 or many of the recent years. So the last thing I want to talk about is regional variability, and we can see strong variability depending on whether you're in Northern, Central, or Southern California. And so this will be the map on the back of the two-pager showing that variability. Again, this is the percent corrosive volume, but you can see that in my summary slides, for the last 15 years, the exposure to conditions beyond those marine life experienced during pre-industrial times averages 15%. Those conditions persisted 19 nearer shore and 35% offshore, which we understand the science behind that. The modeled corrosive volumes are larger and more frequently observed in Northern California. The percent corrosive volume of the water column can be as much as 50% or more in the summer, and it drops dramatically when you go to Central or Southern California. In the future, as ocean acidification progresses, larger volumes of seawater in California will have conditions exceeding what was observed in the pre-industrial area. That suitability of habitat, the availability of that will decline, and stressed organisms, we live in a multi-stressor world, will be exacerbated by increases in temperature and decreases in oxygen, so that stress will increase. So that's what we have learned, and I look forward to any questions folks may have. Thank you.
Well, Jan, thanks so much. A lot of information to unpack there, and great work to build atop the buoy and model data. There is a question for you in the Q&A box from Greg about how those pre-industrial levels were assessed. And so that was, do you want to say just something quickly? And then it sounds like
Sure.
there'll be more information on this available once the indicator's released.
Yeah, Greg. So for the buoys, it was basically the published method that Adrian Sutton did in 2016. For the models, it was a little more involved in that. And I'm not going to speak for the modelers. They each had their method. The existence of the Columbia River made it impossible to use some of the algorithms that are based on salinity. And so anyways, to be continued by Kate and Christina. Sorry.
No, that's a big question and tight timing. So thanks for tackling that. Lastly, we'll hear from Erin Satterthwaite. So Erin's with Cal Coffee and California Sea Grant, and she'll be talking about the development of a biological indicator using Cal Coffee data. Over to you, Erin.
Great, thanks. And thank you so much. I'm super excited to share work on starting to explore biological indicators of ocean acidification and hypoxia using Cal Coffee data. And I just wanted to say I'm extremely grateful for all the people who have contributed to this work, including my collaborators, Todd Martz, Bryce Simmons, and we worked with a team of undergraduate data science students at UCSB who worked on this project as part of their capstone. So following up from what Jan had mentioned, thinking about why biological indicators are needed, they're needed to better understand this relationship between OAH and marine species, and this was reflected in a recommendation from the California OAH monitoring expert panel. And so the key characteristic of biological indicators are that they're sensitive to OAH, that they're ecologically relevant, that they're practical and relatively easy to measure, and ideally have existing data to work from. And so you can see one of the figures that was from this California OAH monitoring expert panel report, looking at some of these various biological indicators. And here within California, we have CalCoffee, which is the longest marine ecosystem observing program in the world and samples the ocean quarterly off of California. And CalCoffee collects physical, chemical, and biological samples and data, including carbonate chemistry measurements. And CalCoffee has the longest time series of direct inorganic carbon observations in the Pacific Ocean. And you can see some of this work was published by Wiley Wolf just a couple of years ago. And also has over 75 years of various zooplankton data. So the goals of what we had worked on were to see if there's co-located carbonate chemistry and zooplankton data from CalCoffee, and if so, assess what the overlap of those looks like between the various data sets, and then to conduct an initial exploratory analysis linking those various measurements. And we examined these 3 zooplankton data sets, which are collected on the CalCoffee platform, and in some cases in partnership with the CCE LTER program, and could have potential for biological indicators for the reasons that are listed below each. So we looked at zooplankton biovolume, taxa-specific zooplankton data, and species-specific krill data. And we found that there was decent overlap, so meaning that data collected at the same time in the same place, both between the carbonate chemistry data and the zooplankton data. And I'll focus on just show you what we looked at related to the zooplankton biovolume data, the one that Maureen showed earlier, because it had the most overlapping data within this 1987 to early 2020 range. So we found a slight correlation between the mean DIC. We looked at many different variables. You can see the GitHub repository link at the bottom, but the mean DIC, which is on the x-axis, and the small plankton concentration, which is on the y-axis. And this consists of all non-gelatinous zooplankton, so excludes things like salps and jellyfish. But we were thinking, okay, this just integrates across all of the taxa of zooplankton, so it'd be nice to look at more species-specific relationships. And those actually might reveal more meaningful patterns. So for this, we've started to explore relationships between carbonate chemistry and particular taxa, so things like pteropods, bryozoans, ostracods, and krill. And we found in some cases that there might be stronger correlations. But we want to do further investigation of these to see in terms of both taxa and life stage. And then we'd also like to think about other ways to represent carbonate chemistry. So kind of as Jan mentioned, thinking about this habitat availability or saturation horizon depth. We also want to look across, we have many different biological data sets within CalCoffee. And then finally, we really want to continue to provide timely information and maintain the carbonate chemistry time series by continuing to collect samples. But we have over 3 years of bottled carbonate chemistry samples that are unanalyzed from CalCoffee due to the ongoing financial challenges associated with sample analysis. And so with that, I would love to hear any questions or thoughts. And we're also really grateful to OPC and Sea Grant for supporting this. So thank you very much.
Thank you so much, Erin. And yes, thank you to our supporters, including OPC, Sea Grant, USIUS. We'll have to end it there. So thanks again to our speakers, our collaborators, and to you all for attending today's webinar. We welcome any feedback, further questions, suggestions on future topics, and a recording of this webinar will be made available on OST's website. A reminder about a survey that's being conducted was just linked in the chat if you would be willing to respond. And stay tuned for an announcement for our next webinar in this series. Thank you again. Have a great day.